interdisciplinary field that applies both the principles of life sciences as well as engineering to develop biological substitutes. So these tissue engineered models are nothing but biological substitutes. So we can use all the engineering principles, physical principles, and most importantly, the biological principles. So we should understand the microphysiology of liver if you want to do for tissue engineering. If we are planning for heart tissue engineering, we can plan for the normal architecture of the heart. Accordingly, we can design our scaffold. Scaffold means a substrate on which we can grow the cells, and that will mimic the functionality of the destined organs, may it be liver, may it be kidney, may it be your heart. So depending upon all those kinds of hopes, many people have been trying, and they have already been successful also to make a lot of tissue engineered models. And the main goals of tissue engineering have been to design reliable in vitro models. Reliable means it should somehow match both the healthy as well as pathological tissues and organs. So why we should do pathological tissues? Sometimes question arises. Suppose we want to study the developmental pattern of a disease, so that we cannot directly study in a human, or directly we cannot kill an animal. Rather, we can make some kind of in vitro models in the culture dish, and we can somehow assess what is going wrong. The molecular pathway, maybe the some metabolic pathways. What is really going wrong? What kinds of tissue architecture is getting damaged when the particular liver or cardiac or your kidney cells are somehow changing their morphology. So when we're making this kind of tissue engineering models, we should take into account they should be reliable. That means they should be matching the micro architecture of the liver, micro architecture of the heart. Okay, that's the first criteria we should take into account when we are planning for liver tissue engineering or any kind of tissue engineering. And we should also somehow overcome the known drawbacks, as I was talking about organ transplantation. So the major issues so far with the organ transplantation has been like donor shortage. Who is going to donate you the organ? No one. No one will be really happy to donate until and unless there is some emergency. So this is a big issue with any kind of organ transplantation. Second criteria, even if someone is donating, that should be well accepted by the patient's body and immune system. So we should always look for those criteria. And most of the surgeons who are doing liver transplantation, they see a lot of failure also. Is because of the graft rejection. So to have a control over those scenarios, they sometimes use a lot of immunosuppressive drugs. So these all are the major criteria people have when they are talking about organ transplantations. So when you are making tissue engineered model, we can have a replacement of this organ transplantation. That is one of the hopes or one of the goals of tissue engineering. Then another thing is we are killing a lot of animals when we are doing all these kinds of research for in vivo testing, a lot of drug testing, a lot of modeling. So all of you are biologists, so you will be knowing very well that we do a lot of animal research, and there is an ethics for that. So when we are doing all these kinds of animal modeling, we are most of the time sacrificing the animal just for one organ, and that is not so much physiologically compatible also. Suppose you want to study a liver disease in mice. Mice may not be having normal alcoholic liver disease. It never drinks alcohol. So it may not be getting exact replica of what is happening in case of fatty liver disease or alcoholic liver disease in humans. So animal models are not the real replica, we can say, in terms of physiology, and of course in terms of the size of organs also. So when we are making these models, we should always look into some kind of limitations that is already in the animal models that should be overcome. So there is something called 3R principle, that it is mostly about replacement, reduction, and refinement. So when we are adopting 3R principle, it is more ethical, more economic, and it is mostly matching to the clinical aspect. Like it can finally go to humans. That's the whole idea of adopting 3R's principle. And we can study a lot of complex phenomena, may it be molecular physiology, molecular pathology, and all kinds of changes that are happening during onset of a particular disease that we are trying to replace. So these are the major goals of tissue engineering. Now we are going to talk about particular liver tissue engineering. Before that, I would like to say that this tissue engineering approach has already been tried long back in our history. As early as 3000 BC, there have been trials of skin grafting in some kind of Sanskrit text that is available in our literature. So these are few landmarks that has been successful in trying for tissue engineering. So they have not only tried the skin grafting, but they have also tried for various other models. So we can think of the early ages that our sages and all kinds of Charaka, Sushruta, whatever those people were doing uh, as early as 3000 BC. Okay, so this tissue engineering, even if it was not coined as tissue engineering, but this principle was already there. So the principle of how they were maintaining these tissues, how they're grafting, how they were maintaining without any kind of infections. So all these things were already evident in our literature. And for the first time in 1987, tissue engineering term was coined, and people started using the already existing polymers that are biodegradable. And they also tried various kinds of bioartificial bladders, then trachea, and many more advancements have been happening. You may be seeing some kind of videos that one mice is having human ear. I am sure many of you would have seen those kind of images where a tissue engineered ear have been grown on a mice. So this is one of the success of tissue engineering, that you can grow human ear on the back of a mice. That is a very successful tissue engineering strategy, that the mice is somehow able to grow human ear. Of course, this is not any kind of disease treatment or any kind of modality that we are talking, but this is just to prove the concept how exactly tissue engineering is successful. So when you talk about principles of tissue engineering, we should look for some important components. Okay, suppose we want to isolate cells from a particular patient, and this particular cell is the one that is going to give rise to our destined tissue. So what we do mostly, we isolate cells from the patient, then grow on some kind of substrate. This is called a scaffold. So the green one that you can see here are seeded onto a porous scaffold. And these porous scaffold may be smaller size depending upon the requirement of a tissue. If it is a bone tissue, we can make bone specific substitute. If our objective is for a corneal tissue, cornea, we can make corneal tissue engineering. So the main important thing is that we have to choose the specific cell from the patient that we are going to somehow make our replica or substitute. So once we get our cells, once we have designed our scaffold, then we allow the addition of growth factors that will somehow modulate the cells to further replicate and take over these empty spaces and mimic the situation somewhat similar to a normal tissue, maybe a bone tissue or something. So this is also called as biomimetics. So we are mimicking a existing tissue, but by using tissue engineering approaches. So for large scale production, we can use bioreactor also. So by adopting all these engineering principles, biological principles, and some kind of polymer chemistry, so by using all these combinations, we can reach to some kind of construct, but this is in vitro. When we are using this for some kind of therapy, we should be very specific. So this is the conventional approach that is very common to any kind of tissue. But when we are talking about models, we always look for these three important components, that is cell, then the scaffold components, and the signaling molecules that is inducing the cell to take the shape of the cell of our interest. Suppose you want to take some primitive cells, like suppose if you have heard of stem cells. So these stem cells are like naive cells, they can give rise to any kind of cells. Yes. So these particular cells can be modulated to become the cells of our interest. So by doing so, we can model the tissue of our interest. That's why it is called tissue remodeling or tissue engineering. So we can collect the cells, put on the scaffold, then grow them in some kind of bioreactors, add the respective chemical signals, they can be cytokines, growth factors, or some kind of inducers, and then we can provide some kind of cues so that they can be somehow terminating to the tissue of our interest. This is the overall approach. Okay, so this is the main principle of tissue engineering I wanted you to introduce. Then now coming to liver tissue engineering particularly, that is the topic of today's theme. So why exactly we need liver tissue engineering? So there have been lot of modalities for liver tissue engineering, particularly there can be some kind of damaged liver because of a drug mediated toxicity. Suppose you are taking a lot of paracetamols all of a sudden. There is acute liver toxicity and mostly that requires some kind of medications and liver will get damaged also. So if you have some kind of transplantation option, then you can do this kind of transplantation. This is one of the modality. Second one is a little bit chronic one, that is alcoholic induced as well as non-alcoholic fatty liver disease, and this is the most common cause of liver disease happening throughout the globe nowadays. Okay, so in this scenario when you talk about this disease and the person is somehow suffering with the liver that is damaged, we should have some kind of modalities to treat those kinds of livers. Okay, so when you are trying for all these kinds of transplantations and all kinds of therapy purpose, we need to have a cell source. So these two are major diseases with the liver apart from hepatotropic infection. This is mostly hepatitis, that is viral hepatitis. Apart from that, people are also having some kind of inborn genetic disorders with the liver. For example, Crigler-Najjar syndrome. So these all compilation of diseases are somehow opening up some kind of opportunity for the surgeons and bioengineers to develop some kind of models so that in a disease condition they can develop and replace also. That's the whole idea of tissue engineering: restore, maintain and improve a damaged tissue. So in these kinds of diseases that I just described, the liver is already damaged, and more importantly the hepatocytes are damaged. So when you want to have some kind of salvation, the current scenario is mostly liver dialysis or liver transplantations. But as I told again, with regard to the issues that we are having for the donor availability as well as the rejection criteria, liver transplantation is not going to solve the sole purpose. And this is a statistic you can see in this particular figure, that how many patients are waiting for liver transplantation. This is the percentage, and this is so, as early as 2007. So now in 2020, the percentage of the waiting list for the liver transplantations for these particular diseases is much more. For example, for the participants who are having hepatitis C, it is 30%. So 30% patients are waiting for liver transplantation because there is no availability of the liver. Who will donate it? So these all are non-cholestatic cirrhosis. So cirrhosis is the complete damaging or end-stage liver failure cases where you have this kind of situation. You can see this is becoming like nutmeg. So cirrhosis is a condition where you have almost extreme damage of the liver. Okay. So similarly, during fatty liver disease also you have a lot of metabolic problems, and you can see a lot of fat granules they are getting accumulated. So this is the disease conditions of the heart, the extreme one being the cirrhotic and somewhat intermediate one becoming the fatty. So in all this scenario, finally they end up in liver carcinoma because they are no more functional and they are getting lot of chemicals or maybe lot of metabolites that are fine tuning them towards liver carcinoma, which is the end stage, and it will be become much more debilitating. So when we are having all these kind of patient specific, patient friendly, and it has already been tried also. Like people, a lot of scientists and surgeons have tried cell-based therapy for this liver regeneration. And in this scenario, I would like to inform that they have been successful using stem cell therapy, and they have taken it further, mostly for organ modeling. That is, they have taken from bench to bedside. That means they have tried all this thing in the lab, and they have also been successful to some extent in treating some of the patients. Okay, although it is not ethically allowed directly to use some kind of therapies that are from that are taken mostly from the animals, like pig models. You may be knowing some kind of stories or some kind of news that mostly take the people take some kind of livers or some tissues from another organism and they put into human. So that is somewhat ethically not allowed. Sometimes it undergoes lot of immune rejections, graft rejections. So those criteria can be somehow recuperated with the human specific liver tissue engineering. Okay, so this is the introduction that I wanted to say about liver tissue engineering, what is the relevance of liver tissue engineering, and why we need it. So in this scenario, important thing is that we can make lot of engineered implantable tissues. We can make either liver seed, we can make some kind of tube like structure, or we can make some kind of liver lobule like structures. So these engineered implantable devices are the future of liver damage therapy. So any kinds of disease that you are having with the liver, now the emerging techniques have been so successful, and they are somewhat matching to the damaged liver, that is either being now the current scenario being with the liver transplantation, or somewhat partial transplantation, or some cell transplantation. These all are having some kind of drawbacks. As I told, immune rejection, availability of the cell, availability of the liver. So all these scenarios, even if they are somewhat ongoing, they are not so much successful, and sometimes patient dies even if it is undergoing transplantation. So much hope lies with these engineered implantable tissues, most of the liver models. And this is what I'm going to talk about in more detail today. So when you talk about this disease scenario, may it be hepatitis, carcinoma, or cirrhosis, they all finally end up in liver failure. So what people have been doing, they use some kind of progenitor cells that finally become mature hepatocytes. When I talk in different countries, but all these animal models, there is a approval from Animal Ethics Committee, there is CPCSE in India. We must get all this animal ethics approval before we can manipulate these animals. Humans is not at all allowed because of a lot of issues that you may be knowing. So when you talk about all this complexity, there is ethical issues, but the basic models, they are not matching to the tissue. So one aspect, there is some kind of ethical acceptance and ease of manipulation, and on the other aspect there is non-resemblance to the humans and complexity to see this particular image. So we should somehow think in such a way that our modeling of the liver tissue somehow should balance both. Both means it should be ethically also allowed, at the same time it should match to the physiological tissue. So we should design our tissue matching the liver in such a way that it should be matching both the criteria, that is ethics part as well as your resemblance part. In measure things about the cell type that is present in the liver, the extracellular matrix of the liver, and how they have been designed. So based on all these drawbacks that we have seen, and also their success, how exactly some of them have been successful, even if they are not ethically allowed, but we can take some inputs from these already existing models to try some kind of liver tissue engineering strategy. And that I have tried during my PhD and post PhD works so far. So what is very important is about three important things for the liver tissue engineering. First one is cellular component. Second one is biomaterial component of the liver, that is nothing but your extracellular matrix. And third one is bioengineering strategy. So I have somehow tried to optimize all three components during my works in the last 8 to 10 years. So before modeling the liver disease, we must know all these individual components in much better way. First one is cellular component. What are the natural liver lobule possess? So what kind of cells are present in a natural liver lobule? We must somehow mimic this structure when we are making the liver model. So this is nothing but your normal liver lobule, and it has got your bile duct, it has got hepatic arteriole, it has got endothelial cells, it has got Kupffer cells, and majority of them are your hepatocytes. And the arrangement is also very much crucial, how they are arranged. They are not present somewhat like a spheroid or ball. They are present in some specific architecture. You can see here, they are present somewhat like a disc. So when you are making our tissue, we should match these criteria also. Not only the cell types present, but also the architecture we should somehow mimic. Apart from this, Not only cells are present in a liver, we have also important biomaterial components that are modulating these cells, particularly the liver extracellular matrix. Among them, the major extracellular matrix of liver are collagen, fibronectin, laminin, and there are some other accessory biomaterials. So you also should take into account these biomaterial components. What are the extracellular matrix present in this liver? And when you put our cell to this liver extracellular matrix, they should be happy. That means they should be matching the natural microenvironment of liver. Okay, so these two criteria are very important. Now, third criteria, which is very, very crucial, how we can position these cells and extracellular matrix in our model. For that, we must know about different categories of bioengineering strategies. People have tried about spheroid culture, organ on chip platforms, somewhat like sandwich cultures. And I'm going to talk also the advanced strategies that are available. And we have also adopted using microfluidics, layer by layer, lab on chip. And nowadays people are talking about 3D bioprinting. So all these bioengineering strategies are also very crucial. How we are positioning the specific cells and the biomaterial components. Okay. So with this, let's talk one by one each component, and I will also give my data that I have been these endothelial cells are also very much important. They mediate all kind of transport activities, cellular extravasation, and lot of scavenging roles. Apart from that, we have some of the phagocytic cells like copper cells, biliary epithelial cells, dendritic cells. So these are somewhat minor cells. Okay, so their presence is of course important, but the majority of the cells must be your hepatocytes and endothelial cells. So when we are designing our liver, we must take into account their composition, percentage, and architecture. All these three things have to be taken into account, and we should position that. So in this regard, there have been some kind of literature available that how exactly the major component, that is hepatocytes, have been tried for liver tissue approaches. Okay, so mostly what people do, they can either use mature hepatocytes, they can directly use hepatocytes. But since there are issues with the proliferation of adult hepatocytes or normal mature hepatocytes, it is always preferable to use some kind of precursor cells. They may be primary cells which can be reprogrammed. This reprogramming can be done by multiple mechanisms, and most of you may be knowing the Nobel Prize by Yamanaka, who did some kind of immune reprogramming and got the Nobel Prize with the induced pluripotent stem cells. So this is one of the breakthrough. So you can use a primary cell, maybe a skin cell also. So from the patient, you can take a skin cell and you can use the reprogramming approach to generate induced pluripotent stem cell, and that can be further differentiated to become hepatocytes. So this is exactly what I was talking about. You can take from same patient a particular cell, may not be hepatocytes. You can take the skin cell from a patient and you can somehow fine tune this particular cell to become hepatocyte by using induced pluripotent stem cell approach. And this particular breakthrough, all of you know how exactly the Japanese scientist Yamanaka got the Nobel Prize. And this is very much useful for therapy. So hepatocyte can be generated using all this programming. Okay, so this is one approach. Another approach is you can use a primary cell that can be directed directly towards becoming hepatocyte. And I have used some kind of bone marrow primary cells to become hepatocyte. I will show you my data after this. Okay, so this primary cell can also become hepatocyte. The primary cell can be a normal bone marrow stem cell or any kind of primary cell that is not hepatocyte as of now. So when we are putting some inducers, that can become hepatocytes. Okay. Apart from this, there have been a lot of other options, like you can use pancreatic progenitor, some kind of oval cells that you can somehow induce to become hepatocytes. People have already tried different culture condition optimization also to generate hepatocyte. Whole idea is that when you are making a liver that is bioartificial liver, it must be somehow matching to the patient. That means it should not be rejected. So we should take that aspect always into account when we are looking for clinical application. So I have tried somehow using some of the approaches, mostly this mesenchymal stem cell that was derived from human bone marrow. So I was working as a hospital in Chennai that is mostly a cardiac center. So I used to collect bone marrow from patients who are undergoing uh, some kind of open heart surgery. So when they are opening the bone, this particular rib, there is anyway some kind of wastage of bone marrow. So that bone marrow I was collecting from those patients who are having cardiac associated diseases. Okay, so they are somehow undergoing cardiac transplantation. My objective nothing to do with the heart. My objective is to do liver tissue engineering. I was collecting only the bone marrow mesenchymal stem cell from those patients. Okay, so I optimized some kind of hepatogenic condition culture system wherein I somehow induced these mesenchymal stem cells, which are like primary cells. Okay, so these are like precursor cells. They are nothing now. They are something like a naive. So they are something like they do not know what they want to become. Only when we induce them with some kind of inducer, they can become a specific cell type. So they are some kind of reserve cells in the bone marrow. Most of the scenario they become hematopoietic progenitors. If you are talking about, there is another fraction that is hematopoietic stem cell that generates RBCs and many other cell types almost every day. But the second population that I am doing induction is mesenchymal stem cell. So these mesenchymal stem cells are mostly present in the G0 phase of the cell cycle, and they are mostly in the resting stage. Only when you induce them or somehow wake them up, okay, to become a particular lineage, they somehow undergo transformation to that particular lineage. And this process is called as differentiation, cellular differentiation by induction. And I used those particular approach to differentiate liver kind of cells from these bone marrow stem cells. So I optimized a hepatogenic condition culture system containing sera. So from the same patient that were undergoing some kind of therapy or some kind of transplantation, from that patients I somehow collected the serum and I measured a particular cytokine, that is hepatocyte growth factor. And this hepatocyte growth factor is known to induce the liver fat. So I used natural sera from patients who are having cardiac failure associated ischemic liver disease. So naturally my sera that I collected from patients are having hepatocyte growth factor, and that serum I somehow optimized further to generate liver cell types from the mesenchymal stem cells. Okay, so this is the data that that I have optimized. So I somehow used on the day zero I collected the mesenchymal stem cells and I induced them with some kind of endodermal inducers, because mesenchymal stem cells are of mesenchymal origin. If you know about different germ layers, endoderm, mesoderm, and epiderm, these mesenchymal stem cells are of mesodermal origin. But I want the liver cells which are of endodermal origin. So I must do some kind of transformation from mesoderm to endoderm, which is a very tight task. So to allow these kinds of hepatocyte-like cell generation, I almost allowed 28 days of culture, and in between I did this kind of specification. First I expanded the mesenchymal stem cell for more numbers, then I allowed them for endoderm specification by induction with fibroblast growth factor and some other growth factors. So I first allowed them towards endoderm specification. After that I used four different categories of my experimental uh, inducers. First, I use some kind of jaundice sera. That means the sera collected from patients who are having hyperbilirubinemia. That's what I told. So I used 5% sera from patients. As a control, I took from normal persons who are not having any disease. So this is collected from normal donors. So I collected sera from them as a control. And in the third category, I used already reported inducers of liver cell types, that is hepatocyte growth factor, fibroblast growth factor, and nicotinamide. They are definitely inducing mesenchymal stem cell to liver cell types. But my objective is patient-oriented liver tissue engineering, so that finally my cells will go to patients. In the third group, what is happening? These all are chemicals grown by somewhat maybe your Sigma or some other companies. So they are growing these growth factors in a recombinant manner, and they may not be efficient for cellular therapy. So that's why I optimize some kind of sera taken from patients directly. So it can be autologous transplantation later. So this sera and the mesenchymal stem cells are from the same patient, okay, the group 1 and the MSC, they are from the same patient, and I allowed the differentiation up to 28 days, and finally I could see they are becoming mature on day 28. Okay, so these are the cellular phenotypes that I observed by immunofluorescence for specific liver specific markers, and I could see that with the patient sera induction, I could get very interesting expression of albumin, alpha fetoprotein, and cytokeratin 18. So these three markers are very much giving insight that they become liver specific cells after 21 days of induction. So mesenchymal stem cells, this is untreated mesenchymal stem cells. They are just having the nucleus. You can see the nucleus. This is deficient nucleus. And similarly with the normal patients who are not having liver disease or your cardiac disease, and the patients who are having the cardiac disease as well as liver disease, and the sera that I induced, they are somehow modulating the mesenchymal stem cell to become liver specific cell types, which is matching to my positive control data. Okay, so this is one of the proof that I got that these particular mesenchymal stem cells can become liver like cells, of course after 28 days. But there is a possibility that if you are taking some kind of serum that is already enriched with hepatocyte growth factor, and those patient specific sera can induce your particular mesenchymal stem cell to become hepatocyte. This was my inference, and I further confirmed that with some kind of markers that is confirming the mesenchymal to epithelial transition, and mostly I look for Snell and CDH1. These are the markers, and I could somehow find that in the hepatogenic cocktail group, this group, already known chemicals, I could get 32% of transformation. So out of 100 cells of mesenchymal stem cells, 32 cells became hepatocyte-like cells. But when I used my patient sera induction group, I could get 38%, which is a very nice data. Yes. Anyone is asking anything? Okay, so I could get some of the data that was very much interesting, and I could somehow differentiate my mesenchymal stem cells to become hepatocytes. Okay, so with this, I somehow want to wind up my cellular component and switch to the second component, that is biomaterial component. So the biomaterial is very much crucial when you talk about liver tissue models, and this biomaterial must have some of the hallmarks, like it should be biodegradable. Suppose we use a biomaterial to seed our cells. So suppose I have generated my hepatocytes here. They are not quite enough to generate my liver cells, or the sorry, generate my artificial liver, because they are only cells. They are like grown just on the culture dish. They are not matching to the three-dimensional architecture of my liver. So I must seed them on some kind of biomaterials so that it is matching to the three-dimensional nature of my liver, how exactly it is present in the body. I want to make a chunk of liver tissue, not just a cell culture. So I always need to use a biomaterial which is again supporting the cells being bioactive, being biocompatible, being very porous so that all the nutrients will go out and all the uh, other materials will be adjusted out also. Most importantly, you should be having a better mechanical property that is matching to normal liver. Also, my biomaterials should be degradable in a longer run. Okay, so all these kind of features I must take into account when I talk about making my model. So this bioactivity, biocompatibility, porosity, biodegradability, and mechanical properties are very much crucial for designing the liver model. So in this regard, lot of biomaterials, both natural and artificial, have been already tried. Okay, so natural materials like your hyaluronic acid, alginate, collagen, material, cellulose, they have already been tried for liver tissue engineering. Simultaneously, some of the already important artificial, not I cannot say artificial, but synthetic biomaterials like polycaprolactone, polyl lactic acid, they have already been successful. These all are FDA approved polymers. They have already been tried for liver tissue engineering. So with this information, there is also another additional information that we must match the stiffness of liver when you want to make. So the biomaterial that you are designing must match the stiffness. So with the literature, we know that for a normal healthy liver, that a normal person has got the elastic modulus of around 6 kilopascal. If you know about little of physics, there is elastic modulus, which is very much important for stiffness. So what happens when a person that is having healthy liver gets a disease, that is liver disease? So when this person is having normal healthy liver modulus of 6 kilopascal, and with the onset of the disease, for example, suppose it is getting fibrosis, this is becoming 8 kilopascal. That means your particular liver tissue is becoming more stiffer, more stiffer. If you understand the concept stiffness, that means when you are getting some disease, your particular liver is also becoming more stiffer. So it reaches around 12.5 kilopascal when it is becoming cirrhosis. That means it is becoming much and much stiffer with the onset of the disease, and when it is reaching to the stage of carcinoma, it is more than 15 kilopascal. So depending upon this elastic modulus, we can also design our tissue model in such a way that the mechanobiological aspect of the liver is maintained. So when we want to make a healthy liver, our biomaterial must match this 6 kilopascal. Okay. So if it is more than 6, it will somehow match to all these other disease conditions. So accordingly, we can make our particular liver model. Okay. So we now talked about different kinds of biomaterials, different kind of cell types. Now going to the third thing, that is your strategy, how exactly we can employ to these cell types as well as biomaterial types. In this regard, people have already tried different kind of cultures as well as co-cultures. This may be direct culture, this can be indirect cultures. Sometimes people have also tried some kind of three-dimensional environment that is matching to our own body, like they have used hydrogels, they have used various nanofibrous scaffolds, and they somehow have been successful taking all these physical and biochemical cues. Okay, so they have been successful somehow matching all this mechanobiological aspect, the both cellular part and the biomaterial part, matching the liver extracellular matrix thickness. So this is one of the slide that talks about almost everything. That means your cellular part, your biomaterial part, and the engineering part. So when you talk about engineering part, there have been a lot of developments. Okay, and these developments have been with the evolution of lot of engineering principles. Mostly people are talking nowadays about 3D printed liver, 3D printed heart. Okay, so these are different tissue engineering approaches that people have been trying. Maybe your lithography, maybe some kind of microfluidic chips. I'm going to touch upon all of them. Okay, whatever time is possible. It's already 4. I'm going to take another 15 to 20 minutes, and we will try to wind up with that. So third thing is that is your bioengineering strategy for the liver tissue engineering. So in this regard, I would like to touch upon what I have done. Okay. So the first engineering strategy that I used is electrospinning. Okay. So electrospinning is a technique by which we can use high voltage electricity, and if you somehow use a polymer of our interest, they can generate this kind of fibers, and we can make such kind of fibers that are somehow electrospun. So electrospinning is based on a principle wherein we can use a particular setup, and this is a syringe. This is filled with the biomaterial of our choice. This biomaterial can be your polymer, maybe natural biopolymer, maybe artificial biopolymer. So I somehow used synthetic polymers. So we can feed your particular polymer solution into this particular syringe, And when you are connecting this syringe tip to high voltage as high as 10 to 15 kilovolt, okay, and when you allow this particular regulator, that is flow rate regulator, to disperse the polymer solution, because of high voltage exposure, they are somehow spinning on your particular cover sleeves or any kind of surfaces that you are allowing, and you can collect those nanofibers. And those nanofibers are having some important features like specific diameters, they are porous, and they have got some specific thickness. So this is the mechanism that I'm telling in brief, that we can use electrospinning. If you can use understand this word electrical exposure, and it is spinning some kind of nanofibers. So these nanofibers are somewhat matching to the liver extracellular matrix architecture. So this is one of the data that is visualized under scanning electron microscope, and I have used polymer blends, both natural as well as synthetic. This is polyal lactic acid caprolactone and collagen. So I mixed both together and somehow wanted to generate different kinds of fibers. So you can see these fibers, they are of nanofibrous orientation, and they are very highly porous, and they most importantly match the biological tissue, mostly the liver. Okay, so I wanted to somehow make these particular fibers on which I seeded my mesenchymal stem cells. So when I seeded my mesenchymal stem cells and allowed differentiation up to 28 days, I saw that when I grew them on a blend of both natural as well as synthetic polymer, I could see that they are making some kind of three-dimensional liver-like organoids. Okay, so you can see there are a lot of cells that are somehow aggregating, which was not seen only in the collagen or only on the synthetic polymers, nanofibrous scaffold. These all are nanofibrous scaffold. This is nanofibrous scaffold. This is also, but this one is a blend of both synthetic as well as natural. So I could generate somehow the hepatocytes that are aggregated together as a three-dimensional entity. And this was already published in this journal if you want to see more details. And I further confirmed whether they are real hepatic spheroids or something, and I assessed the markers for them, particular alpha protein, albumin, and cytokeratin 18, and I could see that they are expressing all these important markers, and also I tracked how they become from flat cells to an aggregate. Okay, I further use some kind of approach like patient-oriented one. That's what I was talking about the stem cells. So I took bone marrow from patients. At the same time, I took syrup from the patients, and I grew those particular bone marrow stem cells from the patients on polyal lactic acid gelatin scaffold, and I induced them. This is more patient-centric approach, and I could see that they are attaining all kinds of hepatic features. They are hexagonal, and they express all the markers. And this is also published in one journal called Advanced Healthcare Material, which is a reputed journal in biomaterial field. And if you want to go through, you can go for more details. Okay, so this is the first approach that I adopted, that is called electrospinning. Then the second approach for liver tissue engineering that I adopted during my postdoctoral fellowship at Portugal is microfluidics, which is mostly involving engineering principles. So here I wanted to make exact replica of the liver lobule. That means it is having some kind of bilayers, one side liver, then in between you have some kind of endothelial cells, then another liver plate. So this is somewhat matching more like a physiological architecture of liver. In this approach, I used microfluidics. This is a microfluidic chips. And if you can see this video, I used in one component liver cell and in another channel endothelial cell. And I wanted to make a lot of different kinds of balls. These are nothing but some kind of micro particles that are already having my endothelial cells and the liver cells. And this is nothing but a microfluidics. So this is the microfluidic chip that I used to use, and this is the mechanism how exactly it is making different ratios of this particular co-culture system that is having both of my cells. liver cells as well as endothelial cells, and they can make this particular microparticles, which was further confirmed by confocal microscopy. You can see so nice pictures which is having both the endothelial features, that is CD31, as well as hepatic features, that is cytochrome P. So cytochrome P is a drug metabolizing enzyme which was expressed highly in the liver cell types, and I could make these kind of particles very successfully with the microfluidics approach. Simultaneously, I wanted to make also some kind of microfibers, not only microparticles, but also some kind of microfibers using microfluidics approach. And in 1 second, I could somehow generate multiple copies of these kinds of fibers. Okay, so you can see these are like microfibers, and each microfiber I'll show you some image. So this is scheme where you are having in one portion or one compartment the liver cell types, and in another compartment endothelial cell types. So I wanted to generate these kinds of microfibers. This is much more physiologically compatible liver model. Okay, so this is how it is looking. So I optimize them. They are having different ratios. Some are having only red one, that is endothelial cell types. Some are having only blue one, that is hepatic cell types. And I could make different ratios of them. Okay, so by doing so I could have different permutations and combinations of these particular hepatic endothelial interfaces, which can be used for different drug toxicity screening. Okay, so I tested their features, how they are having different ratios for markers of hepatocytes as well as endothelial cells in these fibers, and they were very highly viable without any cell death. This is the viability assay, and I also compared them for some kind of endothelial specific functionality, whether they are real endothelial cell making your endothelial compartment or not. So this is one of the proof that I wanted to show, that I could be successful if I would have adopted much more interesting approaches like zonation and all. Somehow or other, I could generate anyway these kinds of models that are useful for high throughput toxicity screening. Then third approach is lab on chip approaches for 3D liver models, wherein you can make different lab on chip. The previous one was I used microchips, that is microfluidics. This is little different from that, an advanced technique called lab on chip approach. So wherein you can make the cell already on the chip. In the previous case, I was generating these products using the chip, but here you can grow the cells directly on the chip. This is lab on chip. Okay, so by this approach, you can make all your individual cells like copper cells, hepatocytes, stellate cells, and endothelial cells directly on the chip. You can grow them and you can do the toxicity screening. Okay, so this is lab on chip approach. And the fourth one is layer by layer approach, where we wanted to make exact organ of liver. So what we did, this is somewhat like your miniature liver that you can see with mice. Okay, and these are from some other organisms that has already been reported in literature. So they are having individual lobes. So we wanted to make organ regeneration. Previously, I was talking about some kind of tissue, only liver tissue regeneration. But next, we wanted to make whole organ engineering. That means we wanted to make all these individual lobes of a liver. So this was done by something called layer by layer approach. We used biomaterials like collagen and chitosan, and we encapsulated them with the liver cells, and we somehow use the concept called polyelectrolyte complexation. That means this collagen, alginate, and chitosan, because of their charge, they can somehow become polyelectrolyte complexation. So first, we individually made the collagen bubbles that was encapsulated with our liver cells, and then we somehow made an aggregate, and these aggregates were somehow matching finally to the liver-like structures. Let's see how it is. So this is the one, a zoomed version of the layer by layer approach that I adopted. So it can form multiple liver lobules that are matching like human livers. So this is a human liver based on different portal venous branches. They can be having 8 different segments. Okay, so if you want to somehow segment them on the basis of portal venous branches, so you can have 8 different components of the whole liver. Okay, so we wanted to make these kinds of whole liver organ engineering, and that's how we can somehow become successfully make 8 different lobules based on hepatic segments and their interaction as well as their venous branches, and this is already reported as per Connoid's classification. So if you want to make total venous classifications of the whole liver, there are 8 segments. Okay, so we wanted to make these 8 different segments also, and we could make successfully this by a layer approach, and we could see that these particular lobules are somehow expressing important things, like they could form bile duct like structure. If you can see here, these particular lobules after 28 days could form bile duct like structure with whole construct. And if you can see here, they are expressing also some bile duct specific uh, markers, particularly by phalloidin staining. I could see them after day 14, they already started bile duct formation, which was further confirmed by specific markers, that is your multi-drug resistant proteins, as well as some polarity markers like Jo1, Jona occludens. And we can see here that these specific orientation of the liver cells in this particular organoid are somehow matching to the natural liver tissue, which is one of the breakthrough. And this manuscript is still under preparation. I'm going to submit it very soon. And I wanted to compare how exactly this 3D top-like structure is looking like. So I used a particular tool called Cell Profiler, and you can see here how exactly the liver is oriented in a 3-dimensional fashion. So as of now, what I'm showing from the electrospinning, that is completely 2-dimensional, even if it was making aggregate. Then I optimized about microfluidics. Then I further moved to this layer by layer approach, and this is a 3-dimensional orientation. You can see the nuclear organization as well as all the tissues, how they are present in a 3 dimension, and it is very much vital for organ engineering, that is matching to the normal physiological liver. Okay, so this I used by Cell Profiler, and we have become successful in generating this liver disease a model. Then fifth approach people have been trying is direct bioprinting. Okay, so now you have bioprinter. So this bioprinter can generate the whole organ directly, and some Brazilian scientists already have been successful to generate 3D bioprinted liver, and this is already being reported in the literature. So this 3D bioprinted liver is also a modality that can be used for your toxicity screening and many other approaches. Okay, many people have already tried some kind of liver mimicking modules directly using bioprinting. They have used laser bioprinting, and there have been multiple usage of biomaterials like methylated gelatin. Okay, so these are some of the reports that is published in literature, just to showcase that they have been successful in generating 3D printed bioliver, or that is artificial bioliver, which can be used for toxicity screening as well as disease modeling. Okay, so now talking little bit about the liver disease model that has been tried using all these engineering strategies that people have already tried. Fatty liver disease model on chip. So they wanted to model a disease that is fatty liver directly on the chip. So they particularly put the media, particularly put the inducers and the cell types, and they could see that they can somehow mimic the structure, how the pathophysiological development of fatty liver has been happening. Not in the patient, but directly on the chip, they could understand the mechanism. So this is one of the breakthrough that they have already optimized. Also, people have tried to optimize your cirrhosis as well as hepatitis, and they have chosen specific conditions like how exactly the cells behave in steatosis and hepatitis, and they somehow induced with some kind of fatty acids, and they could generate these kinds of models directly on the chip. So this is liver steatosis on chip, and all these pathological liver models have been studied so that they can be easily understood in the patient also. How exactly does this kind of mechanism would have been happening in the patient? If we have developed a model, we can put a drug onto this and you can somehow control this. So this is the on-chip model for disease modeling. Also, people have tried a 3D alcoholic fatty liver disease model on the chip. This I already showed the previous slide. Okay. Also, people have tried non-alcoholic fatty liver disease on the chip. So they have somehow modeled individual conditions of progress from normal liver to the hepatocellular carcinoma, and in between they have somehow model also steatosis, then steatohepatitis and cirrhosis. So how exactly a normal liver cell is becoming hepatocellular carcinoma? They have studied all the mechanisms directly on a chip. So this is one important contribution. So if you have a liver tissue engineering strategy, you can control this. Okay. So these various liver tissue engineering models can be somehow take care of the animal models that is having all the issues like expensive, time consuming, ethics, and not matching to the physiological conditions when you are somehow developing a drug. Okay. So when you are developing hepatotoxic drug, you kill many things. So you can control this. Okay. So there have been a lot of other screening strategies, like you can do microwave chips and many other things that I already talked about different chips, how it can be controlled, and how we can use them for various kind of screening purposes. So these are various chips already available, and one can try this for multiple high throughput screenings. With this, I would like to summarize that using the cellular sources, using the biomaterial components, and an interesting and advanced patient-centric engineering strategy, you can make a reliable 3D liver model. Okay, and we can choose the patient-inspired strategy, like we can take from the patient and we can do somewhat autologous transplantation. An important thing is we can match the stiffness criteria using the mechanobiological aspect, and it should be hepatomimetic biomaterial. Third one is it should be somewhat physiological. If you are trying to make a healthy liver, and suppose you want to make some kind of pathological model of hepatitis or fatty liver, our particular engineering strategy also should match this one. Okay. More importantly, if you are trying for some kind of drug screening, you should adopt a high throughput approach, respecting the 3 R's principle. Okay. So this modeling of the liver can definitely carry out all these kinds of applications. So whole idea is that whatever tissue engineering we do, may it be liver or any kind of organs, this should be patient inspired and patient centric. Now people are talking about precision medicine. It should be precise for a particular patient depending upon the onset of the liver disease. Okay. So using this liver tissue engineering model, we can somehow take this idea from the bench to bedside. That is scope of there is a tremendous scope of translation from laboratory to clinic. With this, I would like to stop my presentation. I would like to thank Department of Biotechnology, Ramadevi University, who have been somehow supporting both mentally and motivating for this. Also, I'd like to thank 3B's Research Group, University of Minho, Portugal. This is the large group where I was working. Okay, I was awarded with a grant called Microliver for as high as 196,000 euros, which is equivalent to 1.5 crore. So this was all the data that I'm showing was successful only because of this grant, where I was the principal investigator. And I would like to thank the funding agencies from Portugal. And now I have got a SCRB startup grant 2019 from SCRB, Department of Science and Technology, and I have hired a JRF who is going to work on liver carcinoma using this electrospinning approach, and we are trying to target liver carcinoma using some of the phytomedical components. Okay, so I would like to thank again the organizer for giving me this opportunity to talk today, and particular thanks goes to Dr. Pratyush and Arpita. So with this, I would like to stop. Thank you.
Thank you so much, sir. It was a very informative session. I hope all the participants get some good knowledge about stem cells and all. Okay, so now we will start shortly at 4:30 p.m. the oral presentation. So participant, be ready with your presentation. We will gonna do the oral presentation for both the groups, session 1 and session 2. So all the participants are requested to be ready with their presentation.
1 minute, Arpita. Uh, uh, thank you very much, sir. Sir, thank you very much for your presentation. Yeah. And any
Okay.
participants, do you have any questions for Dr. Dillip? Then you can put forward your question. Are there any questions? Have you put any questions in the chat box? Sir, there is a question from Abranil Gangopadhyay. How
Okay.
much time does it
Yeah.
take to develop?
Yeah, the question is somewhat like hypothetical again. It is from Abranil Gangopadhyay who is asking, how much time does it take to develop a full-sized liver from induced pluripotent stem cells? Yeah, it depends. Like when you are making a prototype, so induced pluripotent stem cells are the cells that can be derived from any kind of cells. So when you are making induced pluripotent stem cells differentiate